Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Heterologous chromosomes, definition, behavior, and genetic importance. Table of contents. Introduction to heterologous chromosomes. Difference between homologous and heterologous chromosomes. Heterologous chromosomes pairing. Heterologous chromosomes, example, X and Y chromosomes. Heterologous chromosomes and translocation. Significance of heterologous chromosomes. Introduction to heterologous chromosomes. Chromosomes carry genetic information within cells and provide essential instructions for an organism's growth, development, and functioning. In diploid organisms, chromosomes typically exist in pairs, with each pair made up of two homologous chromosomes that carry similar genes. However, not all chromosome pairs are homologous. Some pairs do not share the same gene sequences. These are referred to as heterologous chromosomes. Heterologous chromosomes, also known as non-homologous chromosomes, are chromosome pairs that carry different genes or have genes arranged in different sequences from one another. These chromosomes differ from each other not only in genetic content, but also in their size, shape, and structural characteristics. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Let's explore the difference between homologous and heterologous chromosomes. Homologous chromosomes are chromosome pairs that carry the same gene sequences and are similar in structure, including features like chromosomal arm lengths and the position of the centromere. These chromosomes pair up and exchange genetic material during meiosis I, a crucial step in genetic variation. In contrast, heterologous chromosomes, also known as non-homologous chromosomes, do not contain the same gene sequences. They differ in genetic content, structure, and appearance, and they do not undergo the same type of pairing and recombination during meiosis. A common example of homologous chromosomes would be autosomal pairs, while in males, the X and Y chromosomes represent a heterologous pair. Heterologous chromosomes pairing. During the process of meiosis, chromosomes pair up and undergo recombination to ensure genetic diversity in offspring. Homologous chromosomes, which carry the same genes, can exchange genetic material through a process known as crossover. Heterologous chromosomes, on the other hand, have different gene sequences and therefore lack the necessary homology for precise pairing and crossing over. Due to this difference, heterologous chromosomes do not undergo the same type of pairing and recombination that is observed in homologous chromosomes during meiosis. Instead, heterologous chromosomes behave independently throughout meiosis, each maintaining its separate genetic identity. This lack of pairing and recombination between heterologous chromosomes plays an important role in preserving the distinct genetic characteristics of different chromosome pairs and contributes to overall genetic diversity in offspring. While typically heterologous chromosomes do not engage in pairing and recombination like homologous ones, there are notable exceptions to this pattern across various species. In some cases, certain regions of heterologous chromosomes may exhibit limited homology, allowing for minimal pairing and recombination. For instance, In mammals, the X and Y sex chromosomes are heterologous but do undergo crossing over in small, specific regions. The behavior of chromosomes during meiosis is not uniform across all organisms. It can vary significantly among different species and even among individuals of the same species. Several factors, including genetic distance and sequence similarity, can influence the extent of pairing and recombination that occurs between heterologous chromosomes. Heterologous chromosomes, example, X and Y chromosomes. In the 20/XY sex chromosomal system present in many mammals, including humans, there is a specific region known as the pseudoautosomal region, PAR, found on both the X and Y chromosomes. This PAR contains homologous DNA sequences, despite X and Y being heterologous chromosomes overall. The presence of this region allows the X and Y chromosomes to pair and undergo recombination during meiosis. This unique feature enables limited genetic exchange between the X and Y chromosomes in this specific region, which helps preserve genetic diversity while maintaining the essential functions of the sex chromosomes. The recombination that occurs in the PAR leads to the exchange of alleles within this region of the X and Y chromosomes. However, the majority of the X and Y chromosomes, outside the PAR, do not engage in pairing or crossover because they lack sufficient sequence homology. Heterologous chromosomes and translocation. Heterologous chromosomes are also involved in certain chromosomal abnormalities, particularly translocations. Translocations are structural chromosomal changes that occur when a segment from one chromosome breaks off and reattaches to a different, non-homologous chromosome. These events typically occur between heterologous chromosomes and can significantly disrupt the structure and function of genes. Such disruptions may result in serious genetic disorders and diseases, including various types of cancers. Translocations involving heterologous chromosomes can interfere with gene expression, regulatory regions, or lead to fusion genes, all of which contribute to disease development. Studying these chromosomal translocations is important for understanding the genetic basis of these diseases and plays a crucial role in their diagnosis and potential treatment planning. Significance of heterologous chromosomes. Heterologous chromosomes carry different gene sequences, which play a vital role in contributing to genetic diversity by preserving distinct genetic identities within each chromosome pair. Their inability to undergo typical pairing and recombination ensures the maintenance of unique genetic content across different chromosomes. This genetic distinctiveness enhances an organism's ability to adapt to changing environments and various conditions, supporting evolutionary resilience. In the case of sex chromosomes, the pseudoautosomal regions, PARs, present on the X and Y chromosomes allow for limited pairing and recombination. This limited recombination not only contributes to genetic diversity, but also ensures accurate segregation of sex chromosomes during meiosis. Heterologous chromosomes are also frequently involved in chromosomal abnormalities, such as translocations. Studying these abnormalities aids in understanding the genetic basis of many diseases and disorders, helping researchers and clinicians in diagnosis and therapeutic strategies. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!